Welcome to The Explainer. Today, we're diving deep into the very heart of who we are, our genetic code. We're going to unpack the incredible story of how we learn to read our own biological blueprint. And we're not going to do it like some dry history lesson. Nope, we're going to follow the revolutionary tools that actually unlock the secrets hidden in our DNA.
So, I want you to think of your entire genome as this massive, massive library. And every single cell in your body has a complete copy. For pretty much all of human history, that library was locked shut. But in just the last few decades, we haven't just found the key, we've developed these unbelievable ways to read everything inside, from the titles on the spines right down to the individual letters on the page.
All right, so here's our roadmap for today. We're going to start with just seeing the blueprint. Then we'll figure out how to light up specific genes, and then get into how we actually started reading the code. After that, we'll hit the genomic revolution, see how it's helping us map human diseases, and finally, take a look at the future of all this. Okay, let's dive into part one, seeing the blueprint. Our story really kicks off with our first peek into that genetic library back in the late 1950s, when scientists could finally see the volumes themselves, our chromosomes.
This first major breakthrough came from a field called cytogenetics. For the very first time, we could take a human cell, use a special stain on the chromosomes, and then actually arrange them into this organized picture called a karyotype. It was like finally walking into our genetic library and seeing for the first time that there were exactly 46 books organized neatly into 23 pairs sitting right there on the shelf.
And this had immediate, incredibly powerful results. It showed that sometimes the library just had an extra book. Just by counting, doctors could finally diagnose conditions that had a clear genetic cause. An extra copy of chromosome 21, for example, is what leads to Down syndrome. Suddenly, the mystery behind these conditions was explained by something you could actually see, a simple error in the number of volumes. Now, to get this kind of information before birth, two main procedures were developed. You have amniocentesis, which is done a little later in pregnancy and samples the fluid around the fetus. And then there's chorionic villus sampling, or CVS, which can be done earlier by sampling a tiny piece of the placenta. Both are really powerful tools, but they each come with their own specific timing and their own risks, making it a pretty complex choice for expecting parents. Okay, so counting the books was a revolution. But what if the problem wasn't a whole extra book, but maybe a single missing chapter? The next big leap in technology let us zoom in and find specific sections inside those volumes.
The technique that made this happen is called fluorescence in situ hybridization, or, much easier, FISH. Just think of it like a glowing bookmark. Scientists design a tiny piece of DNA, called a probe, that perfectly matches the genetic chapter they're hunting for, and then they stick a fluorescent tag on it. When they add this probe to a patient's cells, it zips right to its target and lights it up under a microscope.
This was an absolute game changer for diagnosing something called microdeletion syndromes. These are conditions caused by a piece of a chromosome that's missing, but it's so tiny that it's completely invisible on a standard karyotype. I mean, you could count the books all day long, but you'd never know that a critical chapter had been torn out. FISH let us finally see what was once completely invisible.
And the test result? It's beautifully simple. In a healthy person, you'd expect to see two glowing dots, one for each copy of the gene on that chromosome pair. But if someone has a microdeletion, well, one of those copies is just gone. So on the screen, you only see a single dot. That little absence of light tells you everything you need to know.
All right, so far we've counted the books and we've put bookmarks on specific chapters, but what about actually reading the text itself? The next great leap was going from just finding genes to reading the individual letters of the DNA code. The A's, T's, C's, and G's. Now, this didn't happen overnight, but it was a really rapid cascade of technology. First, we had techniques like Southern blotting, which worked, but they were incredibly slow and clunky. Then, in the 1980s, the invention of PCR, the polymerase chain reaction, changed absolutely everything. PCR is basically a genetic copy machine. It lets scientists take a tiny, tiny speck of DNA and amplify it into millions of identical copies.
That gave them enough raw material to finally read the sequence directly using a method called Sanger sequencing.
And this led to a massive paradigm shift in how diagnostics were done. For a whole bunch of genetic diseases, especially the ones caused by a single-letter typo, a mutation in a specific gene, direct sequencing became the gold standard. It wasn't about looking at the book's cover anymore, or even finding a chapter. It was about proofreading the actual text for spelling errors.
So this brings us to Section 4, the genomic revolution. See, all the technologies we've talked about so far looked at one gene or one chromosome at a time, but the modern era brought something truly revolutionary, the ability to read the entire library, every book, every chapter, every single word, all at the same time.
This is the incredible power of what we call next-generation sequencing, or NGS. If Sanger sequencing was reading one little piece of DNA at a time, NGS reads millions of them simultaneously in this massively parallel process. The leap in scale is just staggering. It's like the difference between a monk in a monastery painstakingly copying a single page by hand and a modern printing press churning out an entire library in an afternoon.
But, this incredible, almost unbelievable power brought with it a totally new set of problems. Because when you can read everything, you inevitably find things you weren't looking for, and things you just don't understand.
And here's the big twist. It turns out our human genetic library is filled with typos, unique spellings, and all sorts of little variations. Most of them are totally harmless, but some of them cause disease. NGS uncovered thousands of these little variations that we'd never seen before, and for many of them, we just don't know what they mean yet. They're called variants of uncertain significance, or VUS, and they create a huge dilemma for doctors and for patients. I mean, what do you do when you find a genetic change, but you have no idea if it's dangerous or not?
But let's talk about the good stuff. In Section 5, we see that, despite all those challenges, this journey had an absolutely incredible payoff. All these technologies led to an explosion in our knowledge, letting us map the genetic causes of thousands of diseases with, frankly, breathtaking precision.
The result is this vast and growing list of conditions that we can now diagnose with genetic certainty. Syndromes that used to be a total mystery, only defined by their physical signs, can now be pinned to a specific genetic root cause. From conditions you've probably heard of like Down syndrome, to rarer ones you see here, like Phelan-McDermott or Angelman syndrome.
I mean, the level of precision we've reached is just astounding. We can now connect a specific tweak in the VHL gene directly to the tumors you see in von Hippel-Lindau syndrome, or a change in the FGF3 gene to achondroplasia. This doesn't just give a definitive diagnosis, it helps manage the condition and it opens the door for developing targeted therapies.
Which brings us to our final section, the future of diagnosis. This whole journey has brought us to a brand new frontier. One that's not really defined by what technology can't do, but by these really profound ethical and interpretive questions about what it can do.
You see, our diagnostic power has literally outpaced our understanding. When we scan a person's entire genome, what's our responsibility if we find an incidental finding? Say, a risk for a disease they weren't even being tested for? How do we explain one of those variants of uncertain significance to an anxious new parent? These aren't technical problems anymore. They are deeply, deeply human ones. This whole revolution gave us the ability to read our own instruction manual, and it completely transformed medicine and how we understand ourselves. But the real question, the one that's going to define the next chapter of this whole story, is right here on the screen. We finally learned how to read the book of life. Now, we have to decide which pages we're prepared to turn.